Hi student, here in this video I am going to explain you about the physiological effects of cytokinins which is in the plant growth and development. Now this topic appears small but very important for the NEET. Already 2025 you asked one question and that also I am going to discuss. So many questions can be created for NEET from this cytokinins. And the cytokinin, it used to have special effect on the cytokinesis in the cell division. In the cell division, whenever we are reading this mitosis, that will be the karyokinesis and cytokinesis. Karyokinesis means nucleus will become two nuclei, and next it will be followed by the cytokinesis. That will be affected, the special effect will be shown by the cytokinins. So this is the one of the important. Next is this where it is, how it is formed. That already he given in the discovery of the PGR, plant growth regulators. It is modified as adenine purine. Modified, remember it. It is not the adenine, modified adenine. How it is N6 perpheryl amino purine he has given. That like that he has given, that is, it is a modified adenine purine. And now if you see from where first they isolated from the hearing fish. Hearing means some people are telling what is this? This is organism like that, but it is a fish. Fish means here the fish which is having the big scales, small size, big size. All these things will come under category of hearing fish. So hearing fish sperm by autoclaved. Why he taken hearing fish sperm? Tell me. That is, the sperm used to have the less cytoplasm in the zoology studied, and it is having the big nucleus. Big nucleus means more that whatever compared to the cell size, the DNA content is more, nucleus size is more, DNA content is more. From that DNA, they extracted the cytokinin in the and given the name as they are the kinetin. And in this one, again he has given one more word there, that is autoclaved. That is a hearing fish by autoclaved. What is this autoclaved? In the DNA I told, but in the DNA, there it is contains of the what? Nucleotides, that is sugar, nitrogen base and phosphate. In that sugar, in that nitrogen base, one of this is the purines and pyrimidines. In the purine, that is, it is the adenine and guanine. Adenine is required. For that, they have done the autoclave. Autoclaved means high pressure and high temperature. High pressure and high temperature up to 115, containing steam, water vapor will be sent on the DNA of the hearing fish sperm. Then that adenine will be modified into the cytokinin that they given the name as kinetin. This is the explanation. Remember it. So derived from the hearing fish sperm by autoclave. Why the sperm only they taken also I explained. Remember it. And next will then the scientists tried to find out naturally appearing any in the plant, because cytokinins are having the effect on the cell division of the cytokinesis in the plants. So such type of things are present in the cell or not, for that scientists tried a lot of. Finally, they came to know and separated that cytokinin isolated from the coconut milk and kernels of corn kernels. Why they selected that? That is also important. A lot of information is there. That what is the corn kernel and coconut milk? They are the monocots. Remember it. Monocots are having lot of endosperm. Endosperm is formed by the triple fusion. Triple fusion means here it is having the 3n number, means DNA is more. That is the important point. Such type of, and corn kernel also they taken the immature, means whenever we are eating that one milky, it is means immature. Such type of milk they taken, or the coconut milk, this is also endosperm 3n number they taken, and they kept the under this autoclave. Then they become the one substance. That substance is nothing, they called it as zeatin. What is the difference between the kinetin and zeatin? Structurally some difference is there, but adenine attachments only. These are the whatever adenine N6 perfluoryl amino purine 1 for 1, and remaining this whatever in the plant, it is a little bit different in the structure, but working is the same. And here in the animal, the sperm whatever we taken that kinetin, that is synthetic, we used to say, because it is not directly used by all the animals, but in the plant it used to take the special effect in the cytokinesis. That is the explanation. So here the zeatin they separated from the corn kernels, unmatured corn kernels and coconut milk. Remember the point. Why it is? Because they are the endosperm. Endosperm is rich endosperm used to present by the triple fusion in the plant, in the monocots. That also because it is having the 3n number. Now this type of the naturally substances with cytokinin is called as a zeatin, and it is observed in the coconut milk and corn kernels. Now here you see where it will be produced. It is participating, as it is participating in the cytokinesis. Wherever the cell division is more, there it will be produced. Where the cell division will be more? In the root apex and shoot apex. That only root and shoot apexes, and shoot buds. Buds also will be having the meristematic cells which are having the cell division. And young fruits whenever it is forming, starting, they also will produce such type of parts only it will be produced. And next what it is, it functions. How it will be? Is a physiological effect. That is, here it is. It helps in the producing new leaves, chloroplasts, new leaves and chloroplasts in leaves. This also will be again one more question. It is indirectly helpful for the photosynthesis. Leaves are important, chloroplasts are important. They will be produced more by the cytokinin, that is the zeatin, we can say. Like that also you can ask in the question in the NEET. Indirectly involving in the photosynthesis, phytohormone is, means that is cytokine. Remember it. And chloroplast and lateral shoot growth. Actually, lateral shoot growth will be suppressed by the auxin. Already we studied. That is called as apical dominancy. That will be rectified by the presence of cytokinin. That is the important point. Cytokinin promotes the lateral shoot growth, and it will be produce the apical dominancy. And one more thing is here, Apical dominance, it is to reduce. And next will be, this is having the cell division also, it is having adventitious root formation. Adventitious roots, you know. Here, remember it, adventitious root formation means here, other than the stem, wherever if you want to get the shoot system, then you will apply the cytokinin. Which will you apply? He will ask in the need. Cytokinin, that will come in the tissue culture. Tissue culture, first they will form by the using of the oxygen. It will form the colors. It in the discovery it came. And next what happens, whenever you applied cytokinins also, then only shoots will come. That is adventitious shoots will be formed by cytokinin. Adventitious roots will be formed with oxygens. Remember it. So here it is lateral growth and overcome the apical. They promote the nutrient mobilization. This he asked in the 2025. Nutrient mobilization will become fast because of this presence of the cytokinins. What is the advantage? Because of this one, it helps in the leaf senses. Remember, he has given clearly sensitivity of the leaves, not the fruits also. Leaf senses will be delayed. The abscission formation will be reduced. It will be delayed. So, senescence will be delayed by which phytohormones already we studied. In the senescence will be delayed in the oxygens only for the early leaves and fruits, and in the gibberellins it will senescence will be delayed, and here also all the three will be reducing the senescence in the different ways. And these are the things which are most important for the need. Now you might have seen photosynthesis island and what is autoclaved, all the things, different things. Even though it is, I am taking less time, but most important things I explained, which must be in your mind. Further, he can ask the question in different ways. Already he asked nutrient mobilization will be done by the which phytohormone, that is the 2025 last year he asked. He can ask. So if you like this video, please give like and subscribe the channel. And in this way, and I already done up to now 19 short videos and 30 long videos in photosynthesis, respiration, and the plant growth and development. If you want to see, and I am going to next enter into the genetics, followed by the molecular basis of inheritance, and then that is then we will see the biotechnology. And then will come all the other things which are the reproduction, the plants, and biological classification, plant kingdom, this whatever the cytology, biomolecules, all the things I will cover it within 2-3 months. So subscribe my channel, and you will get my video immediately. Thank you.